Hello, my name is Rob Murphy, and I'm the Executive Director here at the Havey Institute for Global Health. And I'm the John Philip Fair Professor of Infectious Diseases and Professor of Biomedical Engineering here. Thank you for taking the time to join us this afternoon for our latest installment in the Havey Institute for Global Health IGH seminar series, co-sponsored by the Center for Innovation and Point-of-Care Technologies for HIV/AIDS at Northwestern University, more commonly referred to as CTHAN. The title of today's presentation is A Brief History of Tuberculosis and Recent Advances in Diagnostics. Please take a moment to follow us on Facebook, as well as Twitter, or X, and Instagram at #FSM Global Health. In addition, we welcome you to become an Institute member to receive access to specialized resources and services that support research infrastructure. For more information, please visit our website at globalhealth.northwestern.edu/members. Today, we're very happy to host Dr. Grant Theron from Stellenbosch University. We're old friends with Grant and worked together for many years, and he is going to be leading this seminar event. Grant is a professor of molecular biology and human genetics at Stellenbosch University, a big partner of Northwestern, by the way. He founded and leads the CLIME group, which is Clinical Mycobacteriology and Epidemiology, based at Stellenbosch University in Cape Town, South Africa, the Cape Town area, dedicated to pursuing practical solutions to end tuberculosis via Africa-centered excellence in training. CLIME's research spans diagnostics, transmission, and the microbiome, and has resulted in over 150 publications. This research has directly and indirectly changed World Health Organization, WHO, and national health policies in South Africa, leading to improvements in the standard of care for people infected with TB. He serves on the Innovation Constituency of the StopTB Partnership. There will be time for questions following the presentation. We welcome you to please submit your questions through the Q&A feature, not the chat box, but the Q&A feature that's at the bottom of your screen. Just put your questions in there, and as time permits, I'll be asking Dr. Theron the questions. So now, Dr. Theron, I turn it over to you, and thank you very much for joining us today.
Thank you very much, Dr. Murphy, for the kind invitation to present today. It's a real pleasure. So I'll be talking today about a history of tuberculosis for those of you who are not intimately familiar with the disease, and I'll be focusing down on some recent research, specifically on TB diagnostics, as well as some results and projects from our collaboration with CTHAN and Northwestern University. So as Rob mentioned, I'm based in the city of Cape Town in South Africa, and we pretty much exclusively focus on TB. And the reason for this is that TB is the single biggest cause of death out of any other disease in human history. TB has probably killed over 1 billion people in the history of mankind, much more so than other infectious causes of death. And the WHO last week released their updated World TB report, and TB is the world's leading infectious cause of death, again, after being briefly overtaken by COVID-19. So there were 1.25 million deaths due to TB last year, and it's the biggest single cause of death amongst people living with HIV. Something that we are only beginning to understand is the role of TB in AMR resistance. Not just drug-resistant TB, but treating TB takes many months of broad-spectrum antibiotics, and that itself can generate AMR in microbes other than TB, which is something relatively poorly understood. It's important as we come off the back of, hopefully come off the back of the COVID-19 pandemic, is that COVID-19 in the space of a year or two basically undid over a decade's progress in the fight against TB. TB deaths and diseases had been decreasing for about a dozen years between 2005 and 2019, and really increased for the first time with the advent of COVID. And this was really due to the diversion of existing resources away from TB to COVID-19. And we are still very much feeling the effects of COVID-19, and I'll talk about that a bit more. So this is the standard map showing the incidence per 100,000 of people with TB around the world. And you can see that Africa and Southeast Asia have very high concentrations of TB. And South Africa is particularly badly affected. However, there are many countries in Southern Africa and Central Africa, as well as increasingly in India and China, and places like Peru in South America, where the incidence of TB is extremely high. And depending on your data source, there are reasons to believe in some of these settings, it's as much as 1,000 cases per 100,000 people. So this is very much a global issue, both if you live in these countries and also if you live in low burden TB country, where you have large migrant populations. So Stellenbosch University Medical Campus is based in the city of Cape Town, on the tip of Africa, which is this beautiful city. And it's an extremely wealthy city in some parts, but other parts of it are extremely poor. So this is approximately 20 minutes away from the previous photo that I showed you. And there is this sprawl of informal settlements where a lot of infectious disease transmission occurs. And very close to these communities is where my group works, and we predominantly recruit people affected by TB from the primary care clinics listed there. And we do our research at a facility close by at Stellenbosch University Medical Campus called the BMRI. And this is next to this very large hospital, which is the Stellenbosch University Teaching Hospital, which is a 1,600 bed hospital, which is very large, but it's not even the largest by South African standards. So this just really shows you the scale of the healthcare crisis in South Africa. One of the silver linings that came out of COVID is there was a very rapid digitization of real-time TB records. So this is a real-time dashboard that anyone can view online that breaks down the number of TB cases by district across any part of South Africa, and this is specifically zoomed in on Cape Town. So over the last year, Cape Town itself had approximately 30,000 TB cases. So just to give you some context, the US has no more than 10,000 TB cases a year. So yeah, you have a single city with approximately 3 times the amount of TB in it than potentially the whole of North America. There are hotspots of TB, and importantly, the rate of TB in people who are tested is very high. So about 20% of the people who we test who have classic TB symptoms have a positive bacteriological test result. That is a very high rate. And it's for that reason that although South Africa has a much smaller population, obviously, than places or India or China or Nigeria, it actually has many more TB cases because the prevalence of TB is so high. For those of you who don't know, the TB is caused by a rod-shaped bacterium called Mycobacterium tuberculosis, and this is spread predominantly via the aerosol route, where a patient who is infectious and typically has some pathology in their lung aerosolizes TB, and then that is inhaled by contact, and importantly, that contact progresses to active disease. Now, this is obviously a highly simplistic picture. There are many different factors that influence the events along this chain. And we don't actually have a very good understanding of, for example, why are some people super spreaders of TB and why are some not? Why are some people who are often exposed to TB never infected by it? And why are some people hypersusceptible to it? So these are all really important questions, and unfortunately I don't have time to speak about today, but are all related to the topic of my talk. So this is sort of the natural history of TB infection. So typically there is some form of exposure, and then infection can occur. It doesn't necessarily occur. The bacteria can be eliminated. You can also get immune control of bacteria. And this can result in something called latent TB, which is something that many people in high-burden countries like South Africa have. And latent TB can result in lifelong containment, which it does for the majority of people. But there's about a 10% annual risk, in the absence of HIV, that is, of latent TB transitioning into active TB. And then, of course, this can lead to onward transmission. This is a rather simplistic picture, but it's just to give you an idea of some of the terminology that I will be using. So this is a great piece of research that was recently done, which reviewed the TB survival data from the pre-chemotherapeutic era. And what it really shows is that most people, when they are infected, actually have very minimal symptoms. And some of them develop pathology, which is this subclinical bar, but do not report to be symptomatic. And about 2/3 of all the people who are initially infected actually recover in the absence of symptomology. In the absence of disease. About 1/3 of people overall develop full-on classical clinical TB, and the majority of these people die. However, some of them can spontaneously resolve. This is going back to the era of sanatoria, for example. Some remain perpetually sick, and obviously the majority of them pass away. So TB, it's often said that the different stages of TB etiology in patients is more different within itself and compared to other diseases. So the
Oh, Grant, can you hear me? You froze.
TB and your body successfully contains it. But TB has a huge amount of tricks up its sleeve, right? So in someone who goes on to develop active TB, the macrophage fails to contain the tuberculosis. And yeah, you can see the tuberculosis literally spilling out of the immune cell. And taken together, this really results in a cascade of immune cell death. So for example, if you look at that green macrophage, it's died, it burst, the TB came out, the yellow macrophage swallowed that TB, and it itself then bursts with TB and gets swallowed up by another macrophage, which meets the same fate. So this is really a failure of containment by the immune system of TB. And when that happens, the body's next line of defense is to form these structures called granulomas to physically contain the TB bacilli. Now, these structures can successfully contain TB, and in fact, they contain many different types of lung disease, but TB can also continue to grow within that intragranuloma space. And eventually the bacteria can reach such a high concentration and destroy the tissue within the granuloma to such an extent that the granuloma essentially bursts and TB starts to spill into the respiratory fluids. And these are just some old sort of pathology pictures of advanced tuberculosis. And you can see, you get this literally this cheese-like necrotizing tissue that forms in the lung. And you get these huge areas of cell death, which ultimately kill you. So TB itself is an ancient disease. This is the earliest known example of TB. This is I think it's a mummy from Syria, from the year 2400 BC, who had very distinctive signs of spinal tuberculosis, because TB, although it predominantly occurs in its pulmonary form, it can really affect any part of the body, especially if you're immunosuppressed. So this is one of the reasons why TB is such a challenging disease today, is because it has really coexisted with us for a very long time. And in fact, when you look at the genetic lineage of TB, it very closely matches human migration patterns. So in the pre-modern era, before you had mass transportation, different human populations had different types of TB that had co-evolved with them. And we think that one of the reasons TB continues to be such a problem to this day is because different populations are exposed to distinct lineages of TB that they did not necessarily co-evolve with. And TB is not just about humans, right? So this is just further reinforces how old TB is. If you can think of an animal, I will bet you that it has a version of TB that is evolutionarily adapted to cause a version of tuberculosis in that animal, right? So in South Africa, for example, a big problem is TB in wildlife. So you get elephants, wildebeest with TB. You even get mongooses and seals, all with their own genetically adapted version of TB. So TB really has had a lot of time to learn all sorts of biological tricks to coexist with its host. And the importance of TB, including the route of aerosol transmission, was recognized very early, right? I mean, Aristotle made observations about consumptives or people with TB having bad air and bad breath around them. And if you take that air in, there's something that is disease producing in it. So I had this slide in because it really shows that over the space of approximately 100 years, there was a massive burst of innovation in the TB space, right? Starting with a lot of fundamental research proving that TB was contagious, Robert Koch's discoveries, tuberculin, X-rays, BCG, and anti-TB drugs, as well as multi-drug regimens in the 50s. But this progress did, towards the latter half of the 20th century, begin to stall, right? And it's very interesting to think about why this is the case. So this is the historic number of deaths due to TB in England and Wales. And people often say, well, TB went away in these settings because TB drugs became available. And that is no doubt one factor. But you can see that the rate of TB was the deaths due to TB were already starting to drop fairly dramatically before the advent of TB chemotherapy. So I'm going to take a brief intermission and give you some TB fun facts because I don't want to talk only about doom and gloom and what's bad about TB. So many of you may not know, but these are both things that owe their existence, or at least their popularity, to tuberculosis. We have an early pair of high heels on the left, and we have a men's razor on the right. And shortly after Robert Koch showed that Mycobacterium caused tuberculosis, and tuberculosis had been proved to be transmittable, people panicked, right, about people having long dresses and bringing in spit from the streets and the roads into the houses, and that could be bringing disease into the house. And as a result, women's fashion changed, right? Skirts started to go above the ankles and shoes started to matter, and you started having to have elevated shoes. It was the same for facial hair, right? Facial hair started to be seen as unclean because you could have sputum and saliva in there that carried TB, and razor blades were initially marketed to try and promote hygiene to prevent infectious disease spread. The cowboy hat, or the Stetson hat, some say owes its existence to tuberculosis. He was a hat maker in New York City. He was diagnosed with TB, and this is when there were no treatment options. And he was essentially sent out west to recover in dry air. And when he got there, he realized the hats were terrible, right? The sun beat down on you. It was raining all the time in some parts. And yeah, he subsequently survived and developed this hat for where he had gone to have his TB cured. And my last TB fun fact is really about World War I, right? So the assassination of Archduke Franz Ferdinand sparked World War I. But what many people don't know is that his assassins had all been diagnosed with TB months before and had been given very little time to live. And they had essentially accepted what they knew would be a suicide mission because they knew that their death due to TB was inevitable. And in fact, all of the assassins who were captured alive shortly died of TB anyway in prison. Okay, so let me go back to the science. That's enough fun facts, but I hope it was a welcome divergence. So why has TB control failed in some settings, and how can this be fixed, right? So this is a fascinating slide, which is really a tale of three cities. We have New York City and London and Cape Town. At the beginning of the 20th century, their rate of TB was similar, right? If you compare the red, the green, and the blue lines. But you can see that obviously Cape Town went in the complete opposite direction. And this is not just because of HIV. This data is in HIV negative people only. And it really comes down to standard of living and poverty alleviation. And again, we see the rapid dip in New York and London in terms of the rate of TB happening well before the availability of TB treatment. So, we struggling to end TB because it's a complex multifactorial issue, and unfortunately do not have the time to go in to all aspects and potential reasons why. But I hope I've painted sort of a meta picture, because these other factors are so important when it comes to thinking about new technological solutions to fight TB, because they need to be sensitive to the local context. So I'm going to move forward and focus on TB diagnostics. And really my reason for focusing on TB diagnostics is that it is the weakest link in the TB care cascade, right? We only diagnose probably 2/3 of the annual 10 million TB cases. 3 million people with TB are undiagnosed every year, meaning that they continue to get sick and transmission continues to occur. In those people who were diagnosed with TB, only 2 in 5 were diagnosed using a WHO recommended tool, right? We've had great new technologies. Sure, they have limitations, but we've had excellent new technologies for over a decade. But we are still reliant on old methods that have not been able to penetrate down to all levels of the healthcare system. Clinical diagnosis, which we know is often inaccurate in high burden settings where you have a shortage of trained health workers. Clinical diagnosis is all too common. Only 1 in 3 people receive a definitive bacteriological diagnosis. And the diagnosis of drug-resistant TB is also extremely challenging, right? We completely inadequately test for it. And many people, we don't actually know whether or not they are going on to appropriate or effective treatment. So there was, until the early 2000s, a huge lack of innovation in the TB diagnostic space. The only rapid test we had was the acid-fast bacilli smear microscopy, which was over just several hundred years old. And in many high-burden countries, this was done in a kind of messy room that was filled with stains and had really poor infection control and had all kinds of hazards and problems with it. And it would rely on the diagnosis of bacteria just by the visual examination of a sputum smear slide under a light microscope. And this, of course, has very poor sensitivity for TB, in addition to being very dependent on the level of training of the operator. So many people don't know this, but perhaps the biggest advance in TB diagnosis, at least in this century, actually stemmed from the anthrax scares in the USA in approximately 2005. I mean, I remember when this was happening, and various people had forms of anthrax posted to them through the US postal system. And in response to this, the Department of Defense contracted this company called Cepheid to deploy PCR-based testing, which was very easy to do and could be done in post offices to detect anthrax and to do swab testing for anthrax spores. So this technology was subsequently adapted by a company called Cepheid into really the first highly scaled PCR platform for TB that has seen global adoption. It started to be scaled out in approximately 2011. And there's this animation here showing how it works. Really, its key strength is that in addition to having good sensitivity, it is also 90% automated, right? In other words, once you add a buffer to the sample, you put it in the machine and you walk away. And that is something which, to this day, even technologies that are coming out now struggle to replicate that level of automation and robustness. The test was approved by the WHO, and tens of millions of GeneXpert cartridges have been run worldwide, and it is the standard of care in many countries. But it has a lot of limitations, right? It's not affordable. The equipment is extremely expensive. It can't be done at the point of care. Many countries that invested in it heavily struggle to maintain the instrumentation. So this is really the standard of care at the moment in many places, but it's not good enough. And it has a total hands-on time of approximately 2 minutes and gives you a result in approximately 2 hours. But results can take much longer in some settings if samples need to be transported far away. So one of the interesting things about GeneXpert, which sort of started when I started my research career, is that it started to be really hard to prove that GeneXpert was actually having a positive public health impact, right? It would increase notifications, but in many settings, so many people were treated on empirical grounds anyway. Some people would argue that, sure, this person was now GeneXpert positive, but they would have been empirically treated anyway. And in this systematic review and meta-analysis, and this is data in people with HIV, if you look at the red line, that shows long-term survival of people diagnosed with, the blue line, I should say, shows long-term survival of people diagnosed with smear microscopy, and the red line shows the same for GeneXpert. And there appears to be a small divergence there, but it's a much smaller difference than what we would expect to see. And we haven't fully understood the contextual reasons as to why that is. I think it's crucial to understand those when we think about designing and rolling out new technologies. And just to say that Cepheid, which was purchased a year or two ago by Danaher, was the target of a very successful and a very justified advocacy campaign to reduce the price of their cartridge down to $5 from approximately $20, I think it is. And they made some concessions, but it's really an example of, again, something that was primarily developed with US taxpayer money to fight anthrax being turned into a fairly profitable testing technology. So why have we failed, even with the use of GeneXpert, to really make a dent in TB mortality and TB incidence in the last few years? We are diagnosing more and more TB, but the decreases are not as much as we would expect. And one of the reasons for this, which is a picture that has only really recently started to emerge, or some would say it's actually a very old phenomenon which we've since rediscovered, is that when we look at prevalence survey data, in other words, when everyone gets tested for TB, irrespective of their clinical presentation, we find that about half of bacteriologically positive TB is in people who are not symptomatic. And you can see this is generally true of Africa and Asia. So by only applying TB tests to people who have symptoms, we are automatically excluding many people with tuberculosis who are probably transmitting. So one of the challenges for new tests is that they need to be extendable and useful in people who do not yet meet the threshold for being symptomatic. So there's been renewed vigor, and I don't think the time is ever going to be better to pursue tuberculosis research. And you'll see these are the NTB strategy milestones to end TB by next year, which is obviously not going to happen, sadly. And the progress has been woeful, right? A 50% reduction compared to 2015 was targeted. We've only achieved 8.3%. A 75% reduction in deaths was targeted. We've only achieved a 23% reduction. Importantly, catastrophic costs, even though TB care in many high-burden countries is free, catastrophic costs faced by households due to a loss of employment, caregivers not being able to go to work, etc., is also a major public health problem. So this has really forced a lot of introspection and to take some silver linings from the effect of COVID-19 to try and leverage many of the new technologies that have emerged from that investment. So this is the molecular TB diagnostic pipeline, and it really shows different levels of complexity of tests. So at the bottom row, we have tests that can be done at the point of care. And by point of care in TB, we mean like under a tree, near a village health post, or they can be done near point of care, which is typically at a primary care clinic, or low complexity tests, which require some sort of laboratory facility. And you can see that there are, unfortunately, several gaps still to this day in this pipeline, right? There's generally a very high attrition of products that occurs as you move towards commercialization. And we don't actually have anything really in the near point of care, and certainly in the point of care field, in the clinical validation and the commercialization stages. And the picture is also, unfortunately, a little bit concerning for antigen tests, right? Antigen tests are particularly promising because they can potentially be done on a non-site of disease fluid, such as blood or urine. And we kind of really only have the next generation of urine lamb tests, which is on the bottom here, which I'll talk a bit more about later. But there is a huge need for more technologies to enter late stage commercialization. And the only way for that to happen is for the pipeline to continue to be populated upstream with candidate technologies and biomarkers. So before talking further about specific technologies, I wanted to talk about a concept of diagnostic yield, which at least in the TB field is something that kind of seems obvious when you think about it, but we never really used it to measure the effects of tests. And the way I think about this is, rather than being hyper-focused on sensitivity and specificity, if we think about, for example, COVID-19, potentially the biggest impact that COVID-19 diagnostic testing technology had was not due to highly sensitive, highly specific PCR tests that were done on real-time PCR instruments in sophisticated laboratories. Probably the biggest effect came from rapid assays that could be done at home or near the point of care, even though those tests have suboptimal diagnostic accuracy. And this really refers to favoring tests more and more, even though they have lower sensitivity, if they are more accessible and they can be used in a wider context. And this is just a hypothetical example that was done as part of a WHO exercise. But in this example, you have a test with 90% sensitivity, but it can only access 25% of the population. On the right side, you have a test that has less sensitivity, but it can access more of the population. And the net effect of all this is the green box is bigger, which means you correctly diagnose in more people. And in TB, the classic example we think about, at least in adults, is tests like this on the left. They typically depend on sputum, in other words, fluid coughed up from the lungs. But we know that many people who cannot make sputum have TB and benefit from TB treatment. So this is part of the impetus for focusing on a non-sputum based test. And I think in the past, we've been too harsh on diagnostic technologies because they do not have a certain sensitivity and specificity. And I'm pleased to say that as part of the development guidelines for new TB diagnostics, we are going to be far more forgiving of sensitivity and specificity in favor of having technologies that are much more accessible. So one of the most exciting technologies, and we are hoping that this is positively reviewed by the WHO, is actually a tongue swab for the diagnosis of TB. And what the thinking here is that most of the biomass in the oral cavity is actually in biofilms on papilla on the tongue, right? And the idea is that people with TB are producing small amounts of sputum continuously. They might spit it out, they might swallow it, but that washes over the tongue continuously, building up high concentrations of TB. So this was something which really is a wonderful surprise in the TB field, because it's obviously not sputum dependent. At least you don't need a sputum in that moment, and a tongue swab is extremely easy to collect. So I'll show you some data from various tongue swab diagnostics projects. But the one technology I wanted to focus on, which from publicly available data seems to be the most promising, is a technology out of China called PlusLife, right? And on the left here, we have sensitivity and specificity when done on tongue swabs, achieving very high sensitivity of about 90%. With excellent specificity. So this is in people who are symptomatic, I should just say that. So they do have quite advanced disease. So it's very easy to do. A health worker or a patient can collect a swab. It gets swirled in buffer. It gets put on a lysis machine. The container does not need to be opened again, but it is dropped onto this card device, which is then used for a PCR that lasts about 40 minutes. Now, the other important thing about this type of technology is that it can work with sputum too, right? So if a patient can make sputum, we really want to test it, because sputum is microbiologically very rich, and you can see the performance is excellent there. And then many people, including Andrew at the Desmond Tutu Health Foundation, are starting to look at technologies like this for use in the home. So as the machinery gets more portable, it allows people to potentially be tested at home for tuberculosis. Another very exciting new test on the horizon is a third-generation urine lamb test. Really, the secret here is that many inhibitory compounds are removed, and that allows the signal to be more easily detected. Lamb is part of the Mycobacterium tuberculosis cell wall, and it's excreted in the urine. And this specific product, BP lamb, has this specimen cleaning step, which really amplifies the strength of the lamb signal, which you can see by the gray bars compared to the orange bars. And this translates into pretty good sensitivity and specificity, right? Alea lamb is the first generation lamb test that is standard of care in many parts of the world and is WHO endorsed. But importantly, Alea lamb only really works well in people with immunosuppression, and especially advanced immunosuppression. But BP lamb appears to perform equally well in HIV positive and negative people. And this is tremendously exciting work. So I'm going to move quickly on to digital technologies. So digital technologies, which are essentially specimen-free, hold tremendous promise, right? So computer-aided detection algorithms of chest X-rays, they outperform human readers, and that's been shown very conclusively. And these software solutions, which require a low-dose chest radiography instrument and only a laptop, are starting to be evaluated and widely deployed, especially in Asia. And these are just different algorithms that have been evaluated in this head-to-head comparison by the R2D2 network. And you can see that the area under the curve is fairly similar. The performance is not equivalent. Some perform better than others, but generally this bodes extremely well, especially for high-burden countries where there's a huge lack of expertise to be able to rapidly interpret X-rays. And we can also evaluate X-rays now in comparison with other emerging blood-based tests for TB. So blood-based biomarkers include the expert host response test, which looks at blood transcriptomic signatures. And CRP actually has utility in people with HIV as a triage tool for people who are potentially affected by TB. I'm going to talk about some of our work that's been using AI and cough sounds. So this work started a long time ago with colleagues at Stellenbosch University, and it initially showed that if you ask people with TB to cough, software can tell the difference between a cough that comes from those people and people who are healthy. But I want to tell you a very brief story which shows some of the pitfalls of machine learning, right? So in this particular project, our initial results looked too good to be true. Our area under the curve, or the predictive value of the software, was perfect. It was 1. And when we really drilled down into this, we started to understand that the nurse who was handling the patient, who obviously knew people's TB status beforehand, was only turning on the biosafety control and the ventilation when there was a TB positive patient undergoing the cough aerosol, undergoing the sound sampling. So unfortunately, the student spent many months developing a machine learning algorithm, which basically turned out to be a fan on or off detector. But it really shows how careful you need to be when applying these software solutions. We've subsequently taken this project further. We've collected data from more appropriate types of TB cases, as well as sick controls. And this is currently part of a European Union funded project to put this into an app, which would be used by health workers as people come into the clinic. This is a triage tool, right? Because obviously many things can make your cough sound funny. This is done just to say your cough sounds dodgy. Please proceed to get a confirmatory test. So this is a project called CageTB, which has tremendous potential because most people can make a cough sound. It doesn't have to be a spontaneous cough. So it really gets at that issue of test access that I was talking about earlier. There's a lot of other exciting work in the cough audio space. There was this large multi-center evaluation, but importantly, it showed that different cough audio classifiers are potentially needed for different countries, right? Because when you take a single classifier, you kind of have a one-size-fits-none type paradigm where you have a lot of variation across sites in terms of performance across the countries on the left there. And importantly, cough is also a useful digital biomarker when measured by a smartphone of treatment response. And yeah, you can see cough trajectories over the course of treatment for different types of TB patients. Another very exciting technology that we are starting to look at is vibroacoustics. So this is using a form of point-of-care ultrasound where a minimally trained health worker does a sweep of the torso of a patient, and this generates basically a probability readout that is associated with disease severity. So in addition to diagnosing TB and differentiating these different clinical stages, it's also indicative of disease severity. And this is a very important topic in the TB space, because we think that patients who do not have severe TB probably do not need the full 6 months of treatment, but we don't really know how to identify those people easily. So this kind of tool is very important for informing regimen length as well. Another thing we've done is actually speak to a lot of people affected by TB across different countries. This is part of discrete choice experiments where people are given different scenarios to ask what do they value the most in the test. So on the left there, each column is a specimen type. And what we found overall is that there was not a strong preference for one particular specimen type, at least in the context of this work, which was surprising to us, right? The other thing that people really favored is that many people actually preferred, indicated by the blue, testing in the health facility. But again, this is very country dependent, and we think this might be due to levels of trust in the healthcare system. People obviously preferred a test that was highly accurate. A test with 70% sensitivity was thought to be non-acceptable by patients. And patients obviously preferred a quicker test, as well as a test, of course, that is free. But this is aggregate data, right? It's not differentiated, because different types of people might prefer different things. So for example, as we look into this data in more detail, it's clear, for example, that the type of people who prefer testing to be done at the clinic compared to at the home are actually much sicker clinically, and there's the expectation that they will get a better test if done at the clinic. I want to give a very quick word as I come close to closing on drug susceptibility testing for TB. I mentioned how this is woefully inadequate, right? And even in people who we know have first-line drug-resistant TB, only about half to 2/3 of those people get further drug susceptibility testing. And one of the solutions we've started to develop, and this is work funded by Theron and Northwestern University, is the Credo project, right? And this is really taking a piece of laboratory waste, essentially, that would otherwise be discarded, and trying to use it to diagnose drug-resistant TB. So this is a GeneXpert cartridge, and this cartridge on the right is spent, it's used. The person was positive, and this would ordinarily go in the bin. But we showed that the back of the cartridge can be used to extract diagnostically useful material, which can be used for further drug susceptibility testing. And we've developed a punch-like device that can be used for that purpose. This is important because the main reason people do not get further testing is because another specimen never comes. So this is a way of not adding any burden to patients or clinics. And the other important thing is that because in the case of GeneXpert, the design of the cartridge is fairly universal, you can, in theory, use this for any form of reflex testing on any used GeneXpert cartridge, right? To do further drug susceptibility testing or variant typing, et cetera, et cetera. So I've summarized the diagnostic tools, but they need to be mindful of the context, right? So health systems are very weak. This is the South African TB care cascade. And the bottom line here is that only about 40% of people with TB every year were treated. And the percentages here are the percentage of people who are lost at every step of the care cascade. And diagnostic technologies really need to integrate at each of these steps to prevent care cascade loss. TB research is also chronically underfunded, right? There's about a $5 billion gap in funding for TB research. But just to give you some context, right? COVID received $100 billion in 2020 for TB, for research, right? TB receives less than 1% of that every year, and it kills more people than COVID. And economically, investing in TB research is a no-brainer, right? StopTB have estimated that for every dollar spent, the economy gets about approximately $85 back. So TB is a multifactorial problem, like a lot of diseases, and some people have come up with these sort of Swiss cheese models to show how there's not one panacea to fix TB, and it's certainly not only diagnostics, but diagnostics can be a part of that. And all of these different innovations that are listed along the top there can be used to plug gaps in the TB care cascade. So in conclusion, I hope I've reminded many of you that TB is arguably the most important public health issue in the world, certainly the most important infectious disease. New diagnostic technologies, especially those that are sputum-free and can be done at the point of care, offer promise, but they have to be implemented so that they are robust and they can be done in stressed, resource-limited settings and health systems. And their implementation needs to go hand in hand with strengthening of those health systems. And in summary, although TB is an ancient disease that is chronically underfunded, the technological pipeline and momentum from COVID-19, I think, hopefully provides us with unprecedented opportunities. So with that, I'd like to thank the organizers and the funders. Thank you.
Grant, thank you so much. That was a fantastic presentation. Thanks for bringing us up to date on all those issues. We can put some questions in the chat box and the Q&A box. Excuse me. We have a couple here already. Question one, do patients who recover from TB develop immunity from future infections? And maybe take that even a step further. If a person who's had TB and then all of a sudden they have TB again, is it new TB or is it reactivated TB? A few words on that.
Yeah, great question. So one of the biggest risk factors for getting TB is having had TB before, right? So previous TB is associated with new TB. And in our setting, about 14% of people who've had TB get TB again within 2 years. And that's about 50-50 reinfection and relapse. We don't quite understand the mechanisms behind that. Yeah.
Great. All right, Jerry Cangelosi has a question for you. Great presentation, Grant. What can you tell us about the availability of Plus Life tests inside or outside of China, at least for rural RUO?
So they are in preparation for the WHO review, I think they are being extremely cautious with and being very careful with the manufacturing. So I know stock is very limited. But let's talk, Jerry. Maybe I'll see you at the union meeting. I,
Okay. I have a question. Is there a book that you could recommend that gives a accurate recording of the history of TB? You had quite a timeline.
yes.
It sparked some interest here.
Yeah, its name is not coming to memory now. I think it's by Atul Gawande, who's a US physician scientist. But I don't have a specific name that comes to mind. But I will remember it probably in a few hours from now, and then I'll be happy to share it. Yeah.
Yeah, if you get it, send it to us. We'll put it on the website.
Definitely. Yeah.
Another question. Looking at the pipeline you presented of current and new TB diagnostics, what are the specific barriers that you see to pushing new point-of-care TB tests? How do technological costs, culturally appropriate implementation barriers play in?
So I think, generally speaking, a big problem we have is that technologies are not, or prototypes are not developed from the outset, I should say, without due consideration for where they can potentially be implemented, right? They quite understandably are developed by folks who have not had the opportunity to set foot in, a busy TB clinic in a part of the world that's stricken by HIV, right? So the specific contextual factors, I think there are a lot of issues around stigma to do with blood in some populations. Privacy is an issue, but these are highly regionally specific, right? So in some parts of the world, people don't mind coughing in front of other people to give a sputum. And in Southeast Asia, for example, it's very stigmatized, especially amongst women. So, I mean, they all factor in, and I would really say those kinds of issues need to be on the same status as developing technical features. You need to think about pathways to implementation and those contextual factors.
Question from Sally McFall. Does ultrasound technology detect old infections? Anything with ultrasound?
We don't know, Sally. It's kind of at the proof of concept stage. I don't think we see reduced specificity in people with previous TB. But that's very much an open question.
Okay,
I don't know about. Sorry, go ahead. Yeah.
Jerry Cangelosi has a follow-up question. Yes, I hope to see you at the union meeting. You said that the ratio of relapse and reinfection is approximately 50-50 in your region. How do you distinguish one from the other? Or do you bother?
So it's not programmatically distinguished, but in long-term cohorts, it's using whole genome sequencing. So the isolate from the first episode is sequenced and compared to the isolate from the latest episode. And if there are very few SNPs difference, it's considered conventionally to be relapse.
Dave Kelso asks, are any tests developed that can be used in the townships by trained people, not med techs?
Yeah, I think the urine lamb tests have a lot of potential, right? I think there are, unfortunately, a lot of regulatory hurdles around this. A lot of regulators are not as comfortable as I think they should be with so-called trained lay people doing a test. But we are seeing tests get to the level of, for example, the classic HIV rapid antigen test.
What about low dose X-ray?
So low dose X-ray is used in Southeast Asia by people who, to my understanding, are not medical technicians, but they receive some form of short-term training. And they are people who live in the communities where they work. So Yeah, the technology is definitely there. I think the regulatory space is problematic, at least in Africa. So, for example, those technologies, we're not actually able to use them in South Africa due to outdated legislation at the moment.
And Chad Achenbach says point-of-care TB diagnostics hold the promise of decentralization of testing, but do you have any local efforts to improve linkage to care and TB treatment as well? And how do we close that test and treat loop?
Yeah, so that's so important, right? So, we currently currently about 25% of people who are diagnosed with TB, who we know have TB, never start treatment, right? So Chad, I think the fact that the technology is point of care seems to matter a lot, right? People tend to take a result much more seriously if they see their sample being taken and tested in front of them. And we see that both in the healthcare facility context and with forms of active case finding in households. So even that act seems to sort of engender increased value in the test result. But the technology systems also that are used to manage test results and to notify patients of their test results are, quite frankly, stuck in the dark ages. So I think digitization has huge potential. Yeah.
Sally put in a good general resource for TB, a Nature article. Anybody who wants it, the link is there. And then Dave ends up with, what about AI and reading the chest X-rays?
Yeah, so that's what CAD is, computer-aided detection. Yeah, so that
the percentage, right?
Correct. Yeah. So those are that's really what's unlocked the potential of low dose radiation, is because you don't need a person trained in reading chest X-rays.
All right, we're right at the top of the hour. Grant, thanks again. Thank you very much for sharing your insight and expertise today. The presentation was really incredible. Thank you, audience, for joining us and for your very thoughtful questions. We invite you to join us at our next Havey Institute for Global Health event, Global Health Day, for the keynote address, The New Global Health, Improving Health Systems Through Collaborative Research, presented by Margaret Fruck, Professor of Health Systems at the Harvard T.H. Chan School of Public Health and Director of the Quest Network. The seminar will be held in person in the Baldwin Auditorium in the Lurie Medical Research Center here on campus at Northwestern. And you can register via the QR code here, our newsletter, or social media pages. Again, thank you everyone for joining us today. Thank you again, Grant. A recording of the webinar will be available online and will be shared via our newsletter. And this concludes today's seminar. Thanks, Grant. Really appreciate it.
Thank you very much. Thanks all.